Okay, so today we will discuss about how uh, a recombinant protein is expressed in an organism or in an expression system. Now, before I go any further, I should clarify what is meant by recombinant protein. So, like most of you will cite that famous example of insulin being produced in E. coli as an example of recombinant protein. But perhaps we should learn from the fundamentals that a recombinant protein expression simply means that which is not from its natural setting, meaning if you express an E. coli protein, say beta-galactosidase, from any piece of DNA other than the natural chromosome of bacteria, then that beta-galactosidase is a recombinant protein. So if you clone beta-galactosidase in a plasmid and express from that plasmid in a bacteria, that beta-galactosidase is a recombinant protein. Okay. So now that is out of the way, let us get into some of the nitty-gritties of it. Now, in an so often recombinant expressed protein is of commercial value. Okay, sometimes it's a drug, sometimes it's a laboratory reagent, and so many other things. For all you know, that your detergent also has some recombinant protein nowadays, right? So whenever you are talking about such kind of commercial use at a very high scale, then a change in level of expression, even by 1 to 2%, can actually mean a whole lot of money in terms of business transaction. Therefore, attention is generally paid to the finer details to always ensure that the expression is optimum, okay, like the best possible without compromising the quality of the protein. So yield is certainly one consideration. Sometimes functionality is a consideration. Like certain proteins, for them to be functionally active, they need some post-translational modification, meaning after the protein is made, sometimes it is phosphorylated in a particular manner, sometimes it is glycosylated, sometimes it will have lipid modification, and many other things. And these are not always available in all the systems. So there are multiple different systems for recombinant protein expression. I will come to that in a minute. And then the other aspect is that you want to maximize the protein production without allowing the system to degrade the protein. There are proteases in any of the recombinant expression system that you are talking about. So if the recombinant protein is there in the cell for a long enough time, then some of the protein will be degraded. Therefore, the other feature of recombinant protein expression is often that the production is inducible, meaning you know exactly when the protein production will start, and you allow only a short burst for the protein to be produced, and then you purify it or you isolate it from the host system, so without giving much chance to the system for degrading the protein. So like the most elementary version of it is there in this slide. Say you have a plasmid that has the lac promoter operator system, so that it will be inducible by lactose or non-hydrolyzable variant of lactose. The purpose of using a non-hydrolyzable variant like IPTG is so that the intracellular concentration of lactose analog remains high and steady. Okay, so that will relieve the repression of the lac operator, and the gene downstream that is cloned will be transcribed because the repression is relieved, and then it will be translated. Now, the beta-galactosidase is, say, one such gene. Now, you can replace beta-galactosidase with any other YF of your choice, and that protein can be expressed in bacteria. Here, the example shown is GCSF. GCSF is granulocyte colony stimulating factor. This is a highly valued commercial product. After during cancer treatment, if a cancer patient is given chemotherapy, then often the level of red blood cell, white blood cell, they will go down, and then there are protein drugs to actually stimulate the production of that particular cell type. So GCSF will stimulate production of white blood cell. Similarly, there are protein-based drugs for stimulating production of red blood cell, because these blood cells, they have limited half-life, so they are proliferating cells. So therefore, during chemotherapy or radiotherapy, which kills indiscriminately any proliferating cell type. Of course, it kills cancer cells, but it also kills any other proliferating cell type. See, that's why the patients lose hair, because hair follicles are highly proliferative. Patients suffer from indigestion because the gastric lining, the intestinal lining, which renews every 5 days and therefore highly proliferative, they are affected. And what is also affected is the different blood cell types. So there are therefore protein drugs to stimulate those. So here in this example, it is GCSF, and you can express it in bacteria and isolate it from the bacterial cell. In technical terms, you can purify it from all other proteins present in the bacteria, and then you can sell in the market, right? Of course, not you, like companies can sell in the market. They have very strict regulatory norms that you have to satisfy, but that's how it works. Okay. Now, this is what I was talking about, that depending on what you want, there are different kinds of expression system that is there. Okay. So, there is a bacterial expression system, there is mammalian expression system. See, mammalian expression system is often the choice because, like particularly for drugs, etc. But the problem is that the cells grow much slowly. Bacterial cell doubles every 30 minutes, while these cells double every 40 minutes. And it is a little bit difficult to culture them at a very large volume. Not that it cannot be done, it can be done, but it is not as simple as scaling up bacterial culture. There are also expression systems based in yeast, so you can express a protein in yeast. If you want a protein to be glycosylated, then yeast is a very good system. It can also be scaled up very easily. But one of the biggest problems with yeast is that it has a very thick cell wall, and that can often create problem when you are trying to purify a protein. And then there is an insect expression system, baculovirus expression system. So insect cells are the host, and the transgene is delivered in the backbone of a virus that infects mosquitoes. So the insect cell line is called SF9. SF9 can be infected with the viruses, and the virus genome can be engineered to express your favorite protein as well. So, depending on the particular objective you have with your protein, you may choose any one of these. Sometimes you can pursue all two, three in parallel if you do not have the prior knowledge which will be the best system, and you can choose the best one that works for you. Now let us for the first get ourselves completely familiarized with prokaryotic expression system, bacterial expression system. So what are the features that has to be there in a bacterial expression vector? So an expression vector must ensure that the transgene or your favorite gene is transcribed and translated. So the RNA from the DNA has to be produced, and from the RNA, mRNA, the protein has to be produced. And the plasmid has to replicate, multiply in prokaryotic cells. So for that, you will have an origin of replication. You have to identify the cells that are transformed, that are carrying the plasmid. Therefore, there has to be an antibiotic selection, which is often ampicillin. And then to ensure that your transgene is transcribed, there will have to be a promoter. And sometimes you do use the E. coli promoter itself, like a very strong E. coli promoter. I will come to that in a different example in the vector. But often we use promoters that are of bacteriophage origin, like T3 or T7 promoter. These are not of bacterial origin. Bacteriophage are kind of viruses that infect bacteria. So these are bacteriophage origin promoters. They're very well defined, short, 20 nucleotides. So that is most often used. And then because it has to be translated, if you remember that any sequence of DNA downstream of the promoter will be transcribed. Until and unless, and it will continue to transcribe till you have a terminator. Okay? So that will stop the transcription. But when it is bacteria, for the small ribosomal subunit to be recruited to the mRNA that is transcribed from this plasmid, you need to provide a feature called ribosome binding sequence, or RBS, or Stein-Dalgarno sequence. So that is provided from the vector. And then downstream of the ribosome binding site, you will have the multiple cloning site. So this is where you will clone your gene of interest. This is where you will clone your gene of interest. And then once it is transcribed from here, so all of this will be, say there is a terminator here, all of this will be transcribed. And then in the RNA, there will be the ribosome binding site. And this is where the 40th ribosome will be loaded. This is where the 60th ribosome will be loaded. Sorry, just the 40th ribosome will be loaded. And then it will keep on scanning. Somewhere over here, it will encounter the first ATG. And that's where the large ribosomal subunit will come and recruit. And then the rest will be translated. So you will get the protein coming out of here. So that's how the recombinant protein is produced. Now, there has to be an optimal distance between the ribosome binding site and the translation start site. And there cannot be any ATG between the intended start site and the ribosome binding sequence. So the vector by design will ensure that there is no, the plasmid sequence is such that it will ensure there is no ATG in here. Yeah, there is no ATG in here, but just to ensure that the user is providing an ATG, or the ensuring that the translation starts at the optimal distance, multiple cloning site often will provide an ATG. There are two restriction sites, like very popular restriction sites, ND1 and NCO1, whose recognition sequence includes an ATG. For ND1, whose sequence recognition sequence is CAT-ATG, this is where you have an ATG. For NCO1, whose recognition sequence is CCA-TGG, there is a ATG right here. Okay, so expression vectors will often have one of these two restriction sites in the multiple cloning site, which will ensure that the protein expression starts right from that ATG, and it will be at an optimal distance. And this is coming from the vector itself. Therefore, when you are cloning your gene of interest for expression, you may or may not want to have the ATG, the natural ATG of your gene. You don't really need to have the ATG, okay? Not that it will hurt, but generally speaking, you don't want to, because like you will have two methanins in the beginning, but the terminal methionine is often taken out anyway, so it will not harm the protein in any particular manner. But what you also want to, you don't want, I am not saying that this is never done, but if you can, you should avoid that. If this is your intended protein amino acid sequence, you do not want any other amino acid to be included here. Therefore, if you ensure that the cloning is such that the 3 nucleotide after the natural start codon of the mRNA is in a position so that right after this ATG that is coming from the multiple cloning site is rest of the sequences, then you will produce exactly the protein that you normally produce in the cell. Okay. Now, the most popular prokaryotic expression system is basically a two-component system. What do I mean by two-component system? That there is a particular vector that you use, and there is a particular E. coli host that you use. Okay, and I will. We have talked about it in the last class's discussion, but I will just go through it another time. So, like the host that is most popular here is called BL21DE3. BL21DE3 has two very important characteristics. One is that it is lon protease minus. Lon protease is one of the most notorious high turnover protease in a bacterial cell. So if it is lon protease minus, then the degradation of the recombinant protein will be less. And the other thing is that T7 RNA polymerase is lysogenized, because it is part of the bacterial chromosome. Of course, it is an engineered bacterial strain, E. coli strain, but T7 RNA polymerase is under the lac operator system. So, in this bacterial cell, if you add IPTG, then from the bacterial chromosome, which is engineered, the T7 RNA polymerase will be transcribed. And once it is transcribed, then it will be translated, and then that T7 RNA polymerase will bind to this promoter, and it will then do the transcription and translation. And therefore, the vector has to be such that it is recognized by T7 RNA polymerase. So one of the most common classes of vectors, at least during my time, I do not know what is it now, was PET5A. PET5A is a vector that will have the T7 RNA polymerase binding site, and then that's how it is done. So, so this is what is referred to here. So in this particular case, expression of the recombinant protein in E. coli is inducible. How is it induced? So when you add IPTG, it will induce the transcription of T7 RNA polymerase from the engineered bacterial chromosome. Then that will be translated to make the T7 RNA polymerase protein. It is a single subunit RNA polymerase, unlike the E. coli RNA polymerase, and of course the mammalian RNA polymerase too. Then that RNA polymerase will go and bind to the T7 promoter of PET5A, and transcription and translation of your transgene will happen. And therefore, what you should be aware of is that every time you are using such a system, like a two-component system to express your protein of interest, when you add IPTG, if anything else is induced or not, T7 RNA polymerase will be induced. Even the cell that does not have the, like, that is transformed with the vector, that does not contain the insert of your favorite gene. We, in our parlance, we call it vector control. In in laboratory language, it's called vector control. Sorry, not vector. Sorry, vector control. So even in the vector control bacterial strain, T7 RNA polymerase will be induced. It is around 70 kilodalton. So that protein will be always produced. But in the experimental line, experimental cell, where you have the plasmid and in the multiple cloning site, your gene of interest is cloned, then the 70 kilodalton protein will of course be induced. But then imagine you are trying to express a protein that is 50 kilodalton, that will also be expressed. Okay, so that's what is the bacterial expression system. Now, most of the times, the PET system particularly, it does not offer any selection, the blue-white selection. So you don't get to know whether you have been able to clone the insert in your vector or not. It sometimes poses a little problem. So there have been innovations, and now there are plasmids which can offer you both. It will offer you blue-white selection through here. The multiple cloning site is still within the alpha fragment of LAC-Z, and which is under the control of LAC promoter. So with this RNA polymerase, it will be induced, and you will have the here you will have the T7 ribosome binding site. So if your gene is cloned, then the LAC-Z alpha will no longer be produced. So it will be giving you the blue-white selection, but then it will work under the T7 promoter, and your gene of interest will be expressed. If your gene of interest is not cloned, then under TET promoter, the alpha subunit of LAC-Z will be expressed, and you will get the blue-white selection. So you will know if the cells are blue in presence of IPTG and XGAL, then you know that TET promoter operated and expressed the LAC-Z, and that's why you're getting the blue color. And because you are getting the blue color, that means that this WADF is not interrupted, so you have not been able to clone. So you get the advantage of both. Okay. There is another very popular expression system during our time, and I'm pretty sure a variant of it is still used today. It is called PGX-KG. There are certain differences here. Number one is that here T7 RNA polymerase is not used for expression of the recombinant protein. Instead, a bacterial promoter is used. Okay, so it is called TAC promoter. TAC is an artificial promoter I remember the AC is for LAC, and T I think is tryptophan. I'm certainly not confident, I forgot, but it's a fusion promoter. It has taken the nucleotide sequence of two very strong bacterial promoter and created a created an artificial, very strong bacterial promoter. So here, E. coli RNA polymerase will be recruited. If you remember, E. coli RNA polymerase has 5 different subunits. There is an alpha 2, beta 2, and a sigma. Right? So that RNA polymerase will be recruited here, and the gene will be transcribed. And when the gene is transcribed, first thing that it will transcribe is a gene called GST. GST, I think, is 30 kilodalton or something like that. I'm forgetting. So that will be transcribed first, and then comes your multiple cloning site, where you will clone your gene of interest. So what will come out is a hybrid RNA that will have both the coding sequence for GST, and it will continue onto the coding sequence of your gene. And here also, you will have What you need to therefore think very carefully is that GST will have a reading frame, right? It will start with ATG and these are all in here, and then it will have a reading frame every 3 nucleotides, and then suddenly your protein is say you want this frame to be expressed like CCA, the TGG like this. You have to ensure that there are exactly multiple of 3 nucleotides between your ATG and your target sequence, like your favorite sequence, so that there is no change in the reading frame. Your protein will be translated just as it naturally is, but the only difference is that if this is your whole protein, then if this is your whole protein, then the beginning part will have a little bit of added sequence belonging to GST. GST is glutathione S transferase. So this is done because it helps you isolating the protein from the bacteria. So, bacteria will make few thousand different kinds of proteins. To purify your protein of interest from there can often be a challenge. So, having a GST tag helps purification. So, you can make an artificial column where you will have a chemical called glutathione immobilized here, and that's where this GST will bind, and because your protein is fused to GST, your recombinant protein will bind to this column, and then you pour a buffer. That buffer will have very high concentration of glutathione. So now this GST has two options: binding to the glutathione coming from the column, or binding to the glutathione coming from the buffer. Because in the buffer you have a much higher concentration of glutathione, so this will be replaced by the which we call the mobile phase glutathione, and then the protein will come out out of the column, and you'll be able to purify your protein. But it is easier said than done. It will require certain thinking. I will elaborate it in next few slides. So what is a fusion protein? Fusion protein is a combination of two proteins or polypeptides, and in the context of recombinant DNA, it's a comma-less combination of your protein of interest and a polypeptide tag, which you may or may not be naturally occurring and are from the same species, right? So GST is a bacterial protein, but you can have a mammalian gene like yeah, so no, I misspoke. So GST is there in both prokaryotes and eukaryotes, but it doesn't have to be from the same species of GST that you are fusing. So anyway, that's what is a fusion protein. So why do you need to use a fusion protein? So you can use it for easy detection of the protein. For detecting a protein, you generally need an antibody, and sometimes production of antibody is time consuming, or it's difficult for technical reasons, or simply very expensive. So on the other hand, against most of the tags, you will have commercially available antibody that are relatively more affordable. So that is one, so for easy detection. Sometimes, I will show you a few images in the next 1 or 2 slides. You can fuse it with fluorescent protein like GFP, DS-RED. So your protein will glow in the cell, right? So you will be able to see it. Sometimes you can use it for histology. If you remember, for RNA in situ hybridization, we were using anti-DIG antibody that was alkaline phosphatase tagged. Instead, you can have your, so that is basically an antibody, which is a protein that is tagged with alkaline phosphatase. That is for ease of histology or molecular histology detection. Okay, Western blot again, like because of the presence of commercially available antibody, and then purification. Okay, like as I said, that glutathione transferase or GST is a very common tag for purification purposes. The other tag that is very frequently used is histidine tag. You will have 6 histidine residues before your protein sequence, and that actually chelates nickel or many other bivalent cations. So you can very easily purify this protein by having nickel ions immobilized on a matrix. So this recombinant tagged protein, which has the 6 histidine residues, will bind to the nickel, and then you wash it off, and then eventually you so every other protein will be washed off other than your fusion protein, which is bound to the nickel NTA column, and then you pass a very high concentration of copper sulfate containing column through it, or imidazole containing buffer, and that will then there will be a competition. So imidazole kind of looks like histidine. So imidazole will try to bind the nickel column, and your protein will come out, and you will get it in the out of the column. So these are easy to purify. So when you are trying to clone a fusion protein, like when you are trying to produce a fusion protein, there are several considerations. So I am showing you a vector system that was very popular during our time. And I will explain what it does. So this is the promoter, of course, the origin of replication. This is the antibiotic selection, and this is the T7 RNA polymerase promoter. So here also you see T7 RNA polymerase promoter is used. Therefore, your choice of bacteria will be BL21DE3, because that bacteria produces T7 RNA polymerase. The normal E. coli cell does not produce T7 RNA polymerase. It is not an E. coli endogenous protein. It's a bacteriophage protein. So anyway, so you will have the T7 RNA polymerase promoter, then the ribosome binding site. Right after that, the ATG, just to maintain the optimal distance, and then there are the 6 histidine tags, which is used for protein purification. Then this is express epitope tag, okay, because it's a commercially available vector. So the same company has developed an antibody against this sequence of amino acids. So it is very easy for you to detect your fusion protein. So this is to purify your fusion protein, this is to detect your fusion protein. Now you can understand that this is the multiple cloning site where your gene of interest will be cloned. So between your gene of interest and the, so your gene of interest will be preceded by so many kinds of amino acids. It is possible that sometimes they will interfere with the function of your protein. So this particular vector is very smart in having a feature this enterokinase recognition site. This is a very unique thing. So if you treat this fusion protein in vitro with this protein called enterokinase, that will cleave the protein. Okay, so if this is your fusion protein and this is the enterokinase site, once you treat with enterokinase, it will be cleaved, and you will be having mostly your protein and few amino acids extra here. Okay, so this vector system therefore offers you the advantage of expression under a strong T7 RNA polymerase promoter, purification through 6S histidine tag, an epitope for easy detection of the recombinant protein, and a specific feature of protein sequence that makes it a target of enterokinase to remove all these extra sequence once you no longer need them. Okay, you have purified the protein, you have used this express epitope tag for antibody-based detection on Western blot. So you know that you have a pure protein, and you simply add enterokinase, and everything is gone. Okay, so that's what it is. So this is the detail of the vector sequence. So this is your 6-cysteine tag. See, this is the ATG. Okay, so this is your ribosome binding sequence. Then this is the ATG. After ATG, you see in the same reading frame there are 6 cysteines, and then there is this express epitope tag, and then there is this enterokinase recognition site, and this is the enterokinase cleavage site, and much after that is your multiple cloning site. So within the multiple cloning site, you have this NCO one site. See, this is CCA-TTG. CCA-TGG. So that's where you have your have an ATG. Therefore, if you do the cloning smartly, then you will ensure that the natural, so say this is your protein sequence that you actually want to express in here, and this protein sequence of course will start with an ATG, and then right after that, so there is CCA. You will ensure that the way you clone it, that this CCA will be in the same reading frame as these groups of 3, as you were saying. So here, you know, it is perhaps not going to be possible, because you see this ATG is out of frame from this ATG. So for this purpose, these vectors come in different variants. We are set A, B, C. All these variants do is they will have 1 or 2 extra nucleotides, so that this ATG will be in this reading frame for one purpose, will be in this reading frame for another purpose, and in the reading frame 1, 2, or 3. So that's why these commercially available vectors from the expression, particularly the fusion expression vectors, they come in 3 different reading frames. So A, B, C. Okay. So I hope you understood what I was trying to say. Now, that was the bacterial expression system. Now we will talk about the mammalian expression system. In mammalian cells, unlike bacterial cell, the origin of replication for mammalian cells is not known. It is not defined. So what is used is a viral origin of replication. Most of the time you will see SV40, most often they use SV40 origin of replication. Okay, and then you clone your cDNA, and then you transfect it. So the transfection is basically what you have is a liquid emulsion that will open up the membrane, and the plasmid will go in, and during cell division the cells will again become fine, and the cell membrane will become intact again, but the DNA is now gone inside. And because of this viral origin of replication, it will replicate a little bit in there, and then the protein is expressed from there. Okay, but this is not very efficient. So there is a reason why this is called transient transfection, because though the plasmid will replicate, but there is no guarantee that when this cell that has the plasmid will give 2 daughter cells, there is no guarantee that the plasmid will go to both the daughter cells. So eventually the plasmid actually is diluted out in the plasmid containing cells are diluted out in the population, simply because the centromeric sequence is not at all defined. So therefore these are transient expression, but you get very high level of expression, so you can work with them. This is used routinely in most molecular biology cell culture laboratories, transient transfection. But sometimes you may want a particular protein to be expressed in a mammalian cell forever. So that is called the stable transfection. So how that happens is that those plasmids will have a neomycin resistance cassette. Okay, so these plasmids somehow gets incorporated in the chromosome of the mammalian cell. Okay, how that happens, I really don't know. Maybe some other people would know. And how would you know which are those cells where the chromosome, the plasmid has integrated in the chromosome? You, just like ampicillin resistance, you have this chemical called G418 in the solution, and the cells that are resistant to G418, normal mammalian cells will not be able to grow in presence of G418, but in presence of the neo-resistance cassette, G418 is degraded, and then G418 resistant clones will be there, just like bacteria. Okay, there will be clones. Each of these clones, they originate from one particular cell, and you have no clue where the integration has happened. So often you will characterize few dozens of these clones to isolate that particular clone where the expression is optimal. The expression is what you expect, the level or the location or the subcellular location or whatever it is. So that's how the stable expression is done in mammalian cells. So this is a mammalian expression vector. Here also fusion protein is produced. Now you see here, this is the origin of replication. This is your ampicillin resistance cassette. This is your T7 promoter. All these are needed for the bacterial part of this to happen. Okay, because when you are cloning the gene, you have to amplify those in bacteria. You grow a lot of plasmid that you do in bacteria. So these kind of vectors are called shuttle vectors. Why? Because they can grow both in the bacteria as well as they will express genes in some other system. Sometimes it is yeast, sometimes it is mammalian cells, sometimes it is some other system. So these are the ones, these are the features that are required for growth and selection in bacteria, and sometimes you may have the T7 RNA polymerase for some other purposes. Okay, but then you will have the SV40 origin of replication for replication in mammalian cells, neomycin cassette for selection in mammalian cells. Then this polyadenylation sequence, okay, SV40 polyadenylation sequence, that will ensure that the transcription that is starting from this SV40 promoter enhancer will the neomycin cassette will transcribe and will get terminated. If you remember your transcription understanding, eukaryotic transcription from your undergraduate courses, eukaryotic transcription RNA pol2 falls off near the polyadenylation sequence. So this is that, it is not a poly-A tail, it is a polyadenylation sequence that is recognized by the RNA polymerase 2 and it falls off, and that's the location where eventually polyadenylation will happen. But this is not a series of A, okay, this is a polyadenylation signal. Okay, now those are for the mammalian like the neomycin cassette. And then for the expression of your gene of interest, let us say whatever it is, my favorite protein, BFP2. Okay, that is cloned here. Now that also has to be transcribed. For that transcription, you will have a promoter. This is again a viral origin promoter, cytomegalovirus origin promoter. So that promoter will be there. And then it will be cloned in here. And then to ensure that the transcription of my favorite gene is terminated, so there will be a polyadenylation sequence, okay, like poly-A tail. Now you see that in this particular case, so here between the promoter and the polyadenylation sequence, this whole thing is there. Okay, this whole thing is there between the promoter and the polyadenylation sequence. So you have cloned it. After that, after your gene, so this is the 5'. Why? Because the promoter is here. After your gene, there is this V5 epitope. Again, this is for easy detection of the recombinant protein. And then there is this 6S cysteine tag. Again, for the purification purpose. And then there is a stop codon. So in the other version, I showed you that the vector provides the start codon. Here the vector is not providing a start codon, but it is providing a stop codon. Why? Because after this transcript is made, so the transcript will be made from BMP2 to this entire length, the stop codon, and then it will run into this polyadenylation sequence, and that's where transcription will stop, polyadenylation will happen. So the mRNA that will be produced will have your gene of interest. That will be the largest part, and then there will be the V5 transcript area, and then there will be the 6S cysteine tag, and then there will be the polyadenylation signal, and that's where it will be transcription will be terminated, and then post-transcriptionally poly-A tail will be added. Now it is ready for translation. Then because it is eukaryote, so no such thing as ribosome binding site is needed because it will have its own cap. So that's where the 40S will be recruited. Then say this is where the ATG is. It will scan from the ATG, 60S will join, and then it will scan, and then it will produce the protein. Now, do you realize that your gene of interest will obviously have a stop codon here, right? So if there is a stop codon here, then ribosomes will fall off here. Your this V5 epitope and this 6S cysteine, etc., will not be translated. Therefore, if you are going for a C-terminal fusion protein, you have to ensure that your gene of interest does not come with its stop codon. So the primer that we will design for PCR, the reverse primer, it must be from before the stop codon. Okay, and then you clone it, and then you ensure that where the last amino acid, the last triplet codon is, that will run into in a comma-less manner to the V5 epitope. So designing these kind of expression constructs is not trivial. So in one of the discussion classes, we will discuss by actual example how this kind of cloning is done, how an N-terminal fusion protein cloning is done, and how a C-terminal one is done. In N-terminal, you should not provide, you may not want to provide an ATG, but you will certainly provide the stop codon because you will need to stop the translation. In C-terminal fusion, you certainly will not provide the stop codon, but you are supposed to provide the ATG. Okay, I hope I can make it clear. Okay. Sometimes, particularly in if you are working with Professor Atik or in our labs, we use viral expression systems. Why do you use viral expression systems? Because there are certain cell types which are very difficult to transfect with lipid-based transfection reagents, but they can be infected with a viral particle. And viruses will come with very strong promoter enhancer, so the protein expression level will be very high. But making the viral based expression system was certainly not trivial till few years back. Now it is much easier, but still it is not a cakewalk. Okay, you will have to, in most of the cases, deal with a 3 plasmid system. You will have 3 different plasmids. Okay. One plasmid will be where your gene of interest is cloned. That will have the neomycin resistance cassette, etc., because you will transfect this. But it is a viral expression system, right? So this LTR, long terminal repeats, this is a viral feature, and so you want your RNA. So this is a retroviral expression system. So you want your RNA containing your gene of interest and the neomycin cassette to be packaged in a bacteria, in a virus, right? So like retrovirus means they will have an RNA genome. So you want the entire viral genome, including your gene of interest, to be packaged. For that, you provide a packaging plasmid, which will have many proteins that are important for packaging of the viral particles. And then you will have another plasmid, which will have the viral coat proteins. So within the coat proteins, these are the coat proteins, these are the coat proteins, and then the packaging plasmid will produce several packaging proteins, and that's how your RNA genome will be packaged in a viral particle, and now you can use this to infect a cell. Once it goes inside, the recombinant RNA will be released inside the cell, and it will viral translation and transcription is little strange, like they undergo post-transcriptional processing to make different kinds of RNA after infection of the cell, and then it will get integrated in the genome because it's a retroviral RNA, right? It will get integrated in the genome through reverse transcription, and then it will be translated from there, transcribed and translated from there, but it will be transcribed under the long terminal repeater, which is a very powerful promoter enhancer system. So this is another expression system. Now, how do you use this? Like, say you want to know, what are the places in the cell a particular promoter enhancer is active. I do not like this term just promoter. I prefer to call it promoter enhancer. I will explain it when I will teach transcription. Okay. Now, here, wherever in the organism, what you are looking at is basically a C. elegans, a worm. Wherever the enhancer is active, it will produce GFP in there, and it is green fluorescent. So you can see these entire lines. I think these are neurons. You can see the whole neuron lighting up because of GFP, and everywhere so this is basically a readout of the transcription from the promoter. But if you want to know where the natural protein is localized in the animal, then you make a GFP fusion protein for this particular receptor, okay, the ODR receptor. And then you see that not this entire thing is lighting up. So even though the transcription is being promoted throughout, but the protein is, even if it is translated only here, I do not know exactly what happens, but the protein is accumulated only at the tip. So this is just one example of use of fusion protein. There are many such other uses of fusion protein. Okay, so that's all that I wanted to talk about in terms of expression of recombinant proteins. I would encourage you to go to the internet and study different examples of recombinant protein expression and their use. Of course, we can discuss a few during discussion hours, because I will ask you questions very specifically from here to design terminal fusion protein, C-terminal fusion protein, different use cases in the context of a problem. So those are the things. So you please start to think about how are the different ways recombinant proteins can be, like what are the different reasons you would like to express a recombinant protein? When would you decide to express it as a fusion protein? What are the different use cases? If you are trying to express it as a fusion protein, what are the considerations that must go into it when you are designing the primers, etc.? Okay, so all of these we need to think through. Okay, so with that, we will end today's class. Thank you, and I will see you soon on Monday. Bye.